So the next section of our meeting today is on genetic susceptibility to neuroendocrine tumors, what's known and what's new. And here we're going to be hearing from Dr. William Foulkes, who's based at McGill University. He has been director of the program in cancer genetics from the Department of Oncology and Human Genetics at McGill University since 2001. He has been just recently appointed full professor with the Department of Medicine. He is recipient of many research grants and a very prolific researcher with more than 290 articles published as an author or collaborator. His focus of research is in genetics of cancer, and particularly in the area of hereditary cancer. So it's a privilege to have Dr. William Foulkes with us this afternoon, and we are looking forward to hear to his expertise on this field.
Thank you very much. So, actually takes me back, because when I was living in the UK, those of you probably can tell I wasn't born here, those of you, when I was living in the UK, actually, I did a lot of work on carcinoid tumours. In fact, I spent time at the Royal Marsden, and a lot of the patients there were seen by Dr. Ian Smith, who was a carcinoid expert. And when I ended up at the Hammersmith Hospital, interestingly, again, we had a lot of patients with carcinoid because the radiologist was an expert guy at hitting the liver tumours and infarcting them. This was in the early days in the 90s. So I actually know quite a bit about the treatment of carcinoid, but I'm going to talk today about genetic susceptibility to endocrine tumours. And in fact, carcinoid as a group does not feature remarkably high on the list because, as many of you know, certainly hindgut or midgut carcinoids are not terribly, as it were, genetic. What I mean by genetic is they're not due to a single gene. In a sense, all carcinoids, all cancers are genetic in a broad sense, but that doesn't take us very much further. So when I'm talking about inheritance and genetics, I mean a single factor, like a single mutation in a single gene that can be inherited in a dominant or recessive way, that to a large extent explains the occurrence of the disease. So in that sense, really the foregut carcinoids, that is of the thymus and the bronchus, are much more genetic than the hindgut ones and the midgut ones. So I'm actually not going to talk much about the hindgut and midgut ones because there isn't much to say. It's a very short talk. It's basically sit down now. And the foregut ones, it's MEN1. Thank you very much. Sit down again. So I'm going to focus, because I think Juan asked me to speak for more than 2 minutes, but less than 2 hours, somewhere in between the two, I thought I'd talk a bit about what's known and what's new and focus on things that actually I'm interested in. And perhaps if I'm interested in them, you'll be interested in them too. And if you're not, well, tough. The door is at the back. No. So, okay, so what's known already? What's new, and this is all very new stuff in the last year or two. The good thing about giving a talk about a subject you don't know a huge amount about is that you have to do a lot of reading. And the great thing about reading is you learn something. So learning's great. So the nice thing about my job is every single day I'm learning something I didn't know. I didn't know some of this stuff until about 2 weeks ago, as Maureen would tell you, till last night. Anyway, so we're focusing on the 3 Ps, or in fact, they're all Ps, as you can see: paragangliomas and pheos, pituitary tumours, and pancreatic neuroendocrine tumours, or neoplasia. So some of these slides were meant for a medical audience. So what I'm going to do is, because I think physicians might be interested and other people who read these things on the web may be interested, I'm going to leave the slides as they are, but I'm not going to read out everything on them because some of it is quite technical. But for those who are interested, it's there now and in the future. And for those of you who are not interested, you can just listen and not watch the slides. But I think the first bits are pretty straightforward. What are paragangliomas? Where do they occur? And what's the underlying causes? These are essentially rare tumours of the autonomic nervous system, and it refers to a group of neuroendocrine cells that migrate. Interestingly, they don't start where they end up. Like the like all guys know, their testes don't start off where they end up. They start off in your abdomen and migrate downwards. And similarly, neuroendocrine cells don't start off where they end up. They migrate into organs. That's why the thyroid gland has those medullary cells in the middle, as does the adrenal gland. They didn't start there embryologically. They went there later. So essentially, paraganglial tumors arise from what are called extra-adrenal chromatin cells. These are sort of these special cells that have these endocrine properties and can be associated with parasympathetic or sympathetic ganglia. So these are basically the nervous system. And again, I don't need to go through every feature of these, but just to make the point, there are differences in the features of these types of tumors, whether they occur essentially in the sympathoadrenal axis or in the parasympathetic axis. The sympathoadrenal ones, if you like, are the secretory ones, such as pheochromocytomas that some of you may have heard of, whereas the parasympathetic ones are more the extremely rare carotid body tumors, where you get a tumor actually in the neck, which can be part of syndromes which also include pheochromocytomas. So what are pheochromocytomas? Again, they're catecholamine, so that's the hormones that make your heart race. Catecholamine producing neuroendocrine tumors, which arise from, as I mentioned, mainly from the medulla of the adrenal gland, that I mentioned earlier, at the top of the kidney, and some arise from outside the kidney, extra-adrenal. And in people who have high blood pressure, they are a rare but well-accepted cause of high blood pressure. We have many patients who were investigated, rather like you heard in the last talk, for years, for anxiety. They saw psychiatrists, and it was, they were crazy, or they had problems anyway. And it turned out that in fact it was a pheochromocytoma causing all these sort of fright and flight symptoms. They tend to occur later in life, but the hereditary forms tend to occur younger. And this is a recurring theme. Hereditary tumors tend to occur 10 or 20 years younger than the average age in the general population. And it essentially makes sense. Anything that's an outlier, you have to ask yourself, why are they outliers? Someone gets colon cancer age 20, for sure it isn't because they ate the wrong food. They're too young. It's got to be something genetic. So similarly, if you get two of these things, if you get one on each side, one in each adrenal gland, it can't be chance. That must be genetic. The chance of each one happening is so rare that two puts you right off the scale. So clearly, that's an important factor, but most of them are not found because they had symptoms. They're found because of another reason. They're scoped. Maybe they have a bit of a renal stone or something, and someone does a scan and says, oh, what's this? They're often picked up by accident. Did I do two there? Yeah, I did. Okay, so as I mentioned, some of the symptoms are quite general: hypertension, high blood pressure, racing heart, and the type of symptoms you get when you're about to give a talk, like this one. Metabolic effects, hypoglycemia. You can get diabetes, for example. You get weight loss. Could be misdiagnosed as diabetes. But you can also get, in fact, completely normal blood pressure if you produce a different hormone that doesn't affect the blood pressure. And as you can see here, the average delay between the first symptom and diagnosis is 3 years. The average. And you can imagine some of the outliers could be 10 years or more. So again, differential diagnosis. This is more, again, for the physicians, what kind of things should they be looking for? But I put carcinoid in there because that, obviously is of great interest to the people in the audience. If I can make this thing work, I'll point it out there. I'm not sure there's any point in me using this. I don't know. Does it help? Or not really? If it helps, let me know. If it doesn't, I won't bother. So then there are cardiovascular features I mentioned. There's also, even weirder disease like porphyria, sometimes, which you've probably never heard of, are misdiagnosed, and even factitious. Sometimes people get hold of drugs and use them and mimic the symptoms. So you have to think, and also they could be using cocaine and so on. So you have to ask fairly detailed questions to make sure these aren't the case. What about, now we move on to essentially the bit that that I know the most about, and that's what are the genetic syndromes involved. And the main ones are listed here. Von Hippel-Lindau syndrome, you may have heard of, MEN2, due to RET mutations, rarely NF1, neurofibromatosis, the elephant man, the classic story. Didn't actually have NF1, but that's the story people remember. And paraganglioma syndromes, SDHD, SDHB, etc., and very rarely MEN1, but that's really uncommon. So, taking from the Lancet, a review, what are the clinical features? Again, I'm not going to go through this in too much detail, just to make the point that there are different types of von Hippel-Lindau. Only one type, there are several types, of which one type specifically, type 2, is associated with the pheochromocytoma. And you can see type 2C at the bottom there. Probably is worth pointing this out. Type C, you only get pheochromocytomas. You don't get the other features. So obviously confusing when you think of a syndrome as being mainly in one organ and it occurs only in another, but that's the way VHL can be. It can only produce pheochromocytomas in some individuals. a particular mutation in the south of Germany that seems to do this for some strange reason. Nobody knows why. Of course, it can occur in MEN2, as I mentioned, but there are some varieties of MEN2 that only have the thyroid disease and not the disease outside the thyroid. And there's this condition called cutaneous lichen amyloidosis, extremely uncommon, but can cause enormous problems with itching on the back, and is often misdiagnosed. NF1 and paraganglioma syndromes I'll come on to. I won't talk about NF1 because really it's a whole talk in itself, but paraganglioma syndromes I'm going to focus a little bit on for the next few minutes. So what's the breakdown etiologically, as I mentioned? So most of them actually are not due to any known gene. They are due to some gene, but not one that we know about, or a mixture of several genes perhaps, or many genes. But a small proportion, under a quarter or so, are due to hereditary causes. And you can see that breaks down if you take that little pie and take it down to the bottom. What are they due to? Well, most are due to VHL, and with a minority due to RET, and then the SDH genes. But then what's interesting is what are the differences between these different susceptibilities? They're not all, not everybody's equally likely to have a VHL mutation or a RET mutation. It turns out they're actually quite different. I'll talk a little bit about VHL for a few minutes because it's a rather important gene, and also RET, which some of you know a lot about, but I'll focus on VHL. 2 minutes. So what are the major features of von Hippel-Lindau syndrome? Hemangioblastomas, endolymphatic sac tumors in the brain. So endolymphatic sac tumors are extremely rare, but if they occur, basically you have VHL. They can cause deafness, of course, because they occur in the ear. Extremely rare, though. I mean, they're really, if you have that, you have VHL, as I mentioned. Hemangioblastomas can occur in the eye or in the usually cerebellum, but also in the spinal cord. And the whole variety of visceral phenomena, particularly clear cell renal cancer, the most serious complication apart from the cerebellar hemangioblastomas, and, as I mentioned, pheochromocytomas and pancreatic neuroendocrine tumors, and also cis-adenomas of reproductive organs such as the ovaries and testes. So what about pheochromocytomas in VHL? They occur in about 10 to 20% of patients. They're young, 30 years of old, they often occur at, but they can be multiple and bilateral, and they can also occur outside the adrenal gland. And about 5% are unfortunately malignant, when obviously the prognosis is much more serious, a much more grave situation. What about the pancreatic neuroendocrine tumors? They arise in up to about 20% of individuals. They mainly don't produce any hormones, so they'll be asymptomatic until diagnosed. And you can pick them up on usually on MRI now. Fortunately, as you know, McGill, we have some of the experts in operating on these individuals, so the morbidity of operations is often very, very low. But occasionally they are malignant. And you can predict this a little bit, that certain mutations in VHL are more likely to produce malignant pheos than other mutations. And certainly if they get to be rather large, greater than 3 centimeters, you really want to get them out before they spread. And here's a picture of all three. So the kind of thing that you probably saw earlier with Dr. Matrakos, but the arrows, the blue arrow shows a renal cyst here. The black arrow shows 2 renal cell carcinomas on both sides, and the yellow arrow shows a pancreatic cyst there, without a neuroendocrine tumor, but there could be one there developing later. I'd like to thank my colleague Kathy Tucker from Australia for this slide, and the next one, which I stole in an airport in Coolangatta in the summer when we were waiting for the plane. I said, have you got any slides on VHL? She said, here's a few. So here they are. And you can see this shows you the mean age of onset of the symptoms in individuals, both in the brain here and also visceral. Really quite a young age of onset. You can see some young as 25 being the mean age of onset, and there's 1 year old. But it's also as high as 67. So not having these symptoms or signs in your 50s doesn't necessarily mean you don't have VHL, but having none of them would really make it very, very unlikely. So what about the paraganglioma syndromes? What's really emerged in the last few years is this remarkable set of genes, proteins, which all interact together in actually a quaternary structure. You have these 4 proteins that sit together. And what's sort of amazing about it is that SDHA actually causes a completely different syndrome. It doesn't have any cancer risk at all. In childhood, it causes, when you have 2 copies abnormal, it causes a sort of a metabolic phenotype, and the children present very early in life with acidosis, and they can be very seriously ill. Whereas the other 3, B, C, and D, cause varieties of paragangliomas. You can see B mainly causes pheos, and C never causes pheos. D causes pheos and often carotid body tumours, whereas carotid body tumours are very rarely seen with SDHC. So again, knowing which type of gene you're looking at is going to be important, knowing which disease you're looking at when you want to do mutation analysis, because it costs money and takes time. So SDHD is particularly interesting. Again, I won't go through all this for those of you who are not specifically interested in the biochemistry and so on. But what's interesting is if you inherit this gene from your mother, you don't get the disease. Only if you get it from your father. It's kind of very interesting. In other words, it's what's called imprinting, that the maternal copy is inactivated in everybody. So if you have a mutation on a copy that's inactivated, it doesn't matter. It's hidden. But if that's passed on to a man, in other words, the woman's son, the imprint comes off, and he can pass it on to a daughter or a son. Do you see? So the pattern of inheritance in the family can be very bizarre. No mother passes it on, but men pass it on to daughters. So you have to be aware of that when you're counselling families at risk, because they may think, well, everyone's at risk, and they're not, because they are the offspring of women. What's amazing is that SDHB, same family, same complex, not imprinted. So don't ask me why. I have no idea. And interestingly, SDHB can also cause renal cell cancer and occasionally papillary thyroid cancer. And again, just showing you they're all sort of, interestingly, all the genes I've talked about all kind of sit together. You've got VHL sitting here, you've got SDHD, energy, and mitochondria up here. So they're all kind of working through the same pathway through blood vessel development. Because as you probably could guess, a lot of blood vessels occur around these tumors. It makes operations kind of tricky. So there are specific features of these different genes which have specific features of the pheochromocytomas and specific features elsewhere in the body and different inheritance patterns, or slightly different anyway, which allow people like me to try and guess which gene is involved in an individual who has a pheochromocytoma or a related type of tumor. So if someone, for example, comes to you with a pheochromocytoma and has medullary thyroid cancer, you're pretty sure it's due to RET, not SDHD. Whereas if they present with a malignant extraadrenal pheochromocytoma or paraganglioma, it's probably SDHB and not VHL. So again, this is kind of where having a few facts to your fingertips helps a bit. So what about, there's an algorithm you could go through, you could work people up either medically by doing all these tests, or you could just do the genetic test. And in fact, increasingly, I suspect we'll just go down the genetic testing road, because this is going to get cheaper, whereas this is where it gets more expensive. If you have to do a lot of tests and it costs the same money, why not just do the genetic test first and sort of ask questions afterwards? It feels a bit of a blunderbuss approach. But as genetic testing is getting cheaper and cheaper, and in a paper in Science last week, a company in California sequenced the whole genome of a person, that's the entire, every single base, as it were, for $4,000. So really, that's so cheap that I don't think it's going to be very long before we'll be doing all these genes without really asking too many questions about what really is going on. An alternative approach would be to be a bit smarter and say, well, what's the features in the person, and how does that relate to which genes like to be responsible? And you can work your way through, for example, if it's unilateral and they're under 20 with a pheo, then it's probably VHL. It probably isn't SDHD. If it's bilateral, again, it's probably VHL. No, those are both sides, left and right. If it's a unilateral pheo with no family history, maybe it's SDHB, and so on. You can work your way through this list and sort of try to be a bit more intelligent. And if you present with a pheochromocytoma over 50, it's probably not worth doing any testing if you have no family history, because you hardly ever find anything in these individuals. So who should be referred to people like us in genetics? Well, family history, multiple cases of pheos or paragangliomas at any age. If there's multiple cases, I don't mind if they're over 50 if there's lots of them. Pheos plus medullary thyroid cancer. Anybody with VHL, MEN2 or NF1 diagnosed clinically, I should be seen by us to find a mutation so that the next generation can benefit from a genetic test, so they won't have to go through all this question of are they at risk or not. If you find a mutation, you can test them before they're ill, so they don't get ill, and that's the whole point. I mean, what we do essentially is preventive medicine. Well, that's the goal, is to try to intervene before people get sick. Instead of waiting 10 years, at least you have the anxiety of knowing that your child carries a mutation in a gene that can give them the same problems that you had, but you never had that chance. So you might like to pass that chance on, even though it will come with anxiety. If it's a personal history of pheo under 40, those patients should be referred to us. Anybody with an extra-adrenal tumor should be referred to genetics. Anybody with a tumor in paired organs, with both the paired organs, should be referred. And anybody with a pheo where you think it looks hereditary, something else funny going on, just doesn't quite feel right, you can refer them to genetics. And then you'd ask these general screening questions that you might ask that wouldn't necessarily include cancer. Might be hearing loss, for example, which has been unattributed to a endolymphatic sac tumor or a hemangioblastoma in von Hippel-Lindau syndrome. They may have a relative who had early onset blindness and then died of something else. That could be a sign that, in fact, they had an eye tumor that was misdiagnosed or not diagnosed. And you can imagine working your way through. You could have dysphagia, which might be due to a carotid body tumor, which was, occult and not seen otherwise. Lots of ways to try and just make sure you're not missing things. So what's new in the last I think I've got 5 minutes. Do you have 5 minutes? Is that good? Yeah, a couple of minutes, okay. So what's new, I think just to point out what's new, what's very interesting now is that you don't actually even need genetic testing to diagnose paragangliomas. You can actually just use immunohistochemistry, which is the way where you get the tumor itself and you cut a piece, and all the tumors, anyone's ever had a resection of a tumor, that tumor is kept in a pathology lab, and in good labs they're kept for 10 years at least. And you can go back and take pieces, it's kept in candle wax, and you cut through it and take a piece and add a protein to the top and just see whether it sticks or not. If it sticks or not sticks, that can tell you stuff. So what happens here is you add these proteins, or antibodies to these proteins, directly to the slide, and you can see whether someone actually has a mutation in these genes. In this paper, it was virtually 100% accurate. So that will make life a lot easier. Just do that test first. If the proteins are all normal, they don't have any of these syndromes. They may have something else like VHL, but they don't have SDHB. A new gene has been found. So it's not just SDH A, B, C, D, it's now SDH5. And interestingly, this whole work was based on one single family, which has been knocking around since 1982, obviously for many more years before that, was first described over 25 years ago. And it is a single mutation in this protein which causes this disease. So sometimes you may think you're the only one in the world, but it turns out it can be extremely informative to people who are on the lookout for rare diseases that can tell us an awful lot. And now there's a new gene that no one knew about. This was published just a few weeks ago. And there's another syndrome called Carney-Stratakis syndrome that, in fact, Konstantin Stratakis was in town recently in Montreal, gave a nice lecture, and there's a syndrome where you get both paragangliomas and these very rare gastrointestinal stromal tumors, and they're due to mutations in the same family of genes. So a variety, so you look out for GISTs in individuals with paraganglioma tumors. So in pituitary, I'll finish with some stuff on pituitary adenomas, because although they may not be particularly relevant to most people in the audience, it's very interesting that now, when I was a medical student, where we did a lot of pituitary work, there were really no genes for this, or pretty much no genes. And now there's 4, and these 2 have really been described in the last few years. This is a gene called P27, and this is one called AIP, or aryl hydratase. And you can, sorry, I went the wrong way. So you can see here that these are the characteristics of the 4 genes. There's a nice review here for those who are interested where they talk about these familial pituitary syndromes. You can see that for some genes, particularly this one here, the AIP gene, there are no other features. It's just pituitary adenomas, but they don't always cause these problems. So you could have a family where only 2 or 3 people in a family have these pituitary tumors, but in fact they are due to the same mutation. But it's just not everybody gets the disease. So that could save you screening for all these other things. If they had one of these mutations, you wouldn't worry that they were going to get other problems, and you would save them the trouble of screening. So in pancreatic endocrine neoplasia, just a nice review that for those of you interested that came out in this journal here in 2009, which just summarized all the genetic conditions that predispose to what they call PEN, or pancreatic endocrine neoplasia. And they showed again, as I mentioned earlier, MEN1, VHL, NF1, and the tuberous sclerosis gene, which I didn't really talk about very much, could all be responsible for this condition. So, there are certain features again. If you see multiple pancreatic endocrine neoplasia, then probably VHL is a more likely diagnosis than MEN1, for example. So how can you get to us? You can get to us by this address here, and I'd be happy to take any questions about that. I'd just like to, if I've got a minute, I'd like to finish with a poem, which is kind of relevant. So this is called Thalassa, for the Greeks in the audience. Run out the boat, my broken comrades. Let the old seaweed crack, the surge burgeon oblivious to the last embarkation of the feckless men. Let every adverse force converge. Here we must needs embark again. Run out the sail, my heart-sick comrades. Let each horizon tilt and lurch. You know the worst. Your wills are fickle, your values blurred, your hearts impure, and your past life a ruined church. but let your poison be your cure. Put out to sea, ignoble comrades, whose record shall be noble yet. Butting through the scarps of moving marble, the narwhal dares us to be free. By a high star, our course is set. Our end is life. Put out to sea. Thank you.
Thank you very much. That was excellent, enjoyable talk. Do we have time to take, sorry? Couple of questions. So Maureen says okay. Any questions in the audience?
Okay.
So
Nope, okay, thanks very much.
Dr. Shah has a question.
If you take the inheritance into account, the way you describe about imprinting and lack of it in certain instances, does the penetrance become greater in the instances that you looked at than few percentage of all the cases? So would you be able to link heredity to the disease if you take into account the way the...
Yeah, obviously it's more difficult when there's imprinting present. Or like with AIP, where you have a lot of non-penetrance. In other words, people have the gene but not the disease. It's important to recognize that genetics, a gene and a disease are not one for one, or rarely. I mean, there are some diseases like Huntington's disease, where if you have, or cystic fibrosis, you have the, pretty much you have the gene, you have the disease. Yeah. But for many of these conditions, it's not 100% risk. So in a family, you have to be aware that someone could go their entire life and have no problems. That's my point. If there's imprinting, it's more disguised because the penetrance, penetrance just is a sort of fancy word for what's your risk of getting the disease, if you like, 100% high penetrance, 0 low penetrance. So the point about imprinting is that if you inherit the disease from your mother, your risk of the disease is zero, but the risk of the disease of your children is 50%. So that's an unusual aspect and has to be taken into account when you're counseling people, that it's not a 50-50 transmission, yes or no.
Thank you.
Okay.
Well, thank you very much. So, what's next?